And now, we are delighted to have Dr. Martin Dawes, head of UBC's Department of Family Practice, here this evening. Please join me in giving him a very warm welcome. Thank you.
Thanks, everyone. Welcome, coming out on a rainy night. Why is a family physician standing up with these guys? About 2 years ago, I had a call at the weekend from a patient, and they told me that their 23andMe result, their genetic result, was already in their chart and that I could have a look at it before carrying on the recommendation for therapy. At that stage, I thought, okay, well, genetics may actually be here as opposed to being talked about. And so I've become gradually more and more involved and working out ways to include genetics into family practice. But that's not what we're here tonight to listen to. What we're here for is a smorgasbord, a real capturing of what is going on in genetics, but what is going on really that's going to affect us. What sort of things might we expect to see now and within the next few months and possibly in the next few years, and what has already been done. So before we start off, I want to mention that this is being recorded, and that just so that you know about that. The second part is that to give people enough time for questions, we'd like you to write down the questions as the speakers go through, because they're going to be very quick, 7 minutes each. Okay? So they're really going to race through this huge amount of material. And so during that time that they're speaking, rather than waiting until the end, which will be about 40 minutes or so, if you could write down your questions, try not to write in the manner that I write as a doctor, so that it's legible, because what we're going to do is have a look at them, and then we're going to hand them round to the speakers, and after the speakers are finished, they're going to start answering the questions. So the second part, the second hour, is going to be interaction between the speakers answering the questions that you've written. So please do write, and what we aim to do is, after all the speakers have gone through, to actually come and collect. So we won't disrupt by walking through in between each talk. We'll just go through all the talks back to back, a lot of stuff coming at you, but then a lot of time afterwards for you to have those questions answered. So let's just start a little bit. How many people have had a genetic test? Oh, yeah, I ought to put my hand up. Okay, how many people have had their whole genome tested? Okay, it's coming. And that's a good representation. Actually, you may have had genetic tests and not actually been aware that they're genetic tests. We look for enzyme markers and things like that in the bloodstream. So I would say that in my practice, about 10% of my patients have had a genetic test. Now, we're doing a lot of chromosomal testing at birth, so a lot of the young people have had genetic tests. Just they may not have been told or been informed that actually it was a genetic test. So, it's coming. Some of you already had it. And what we're doing here is, as I say, a smorgasbord, a real look. And our first speaker has actually taken the time, because his wife was giving him such a hard time talking about personalized medicine, to actually write a book about it. And that is over there. I can attest to being readable because I had to read it and make comments on it. But it is actually a very, very readable book. I've just sent it to my father-in-law in the UK, who's 96. And he's now going and looking up what chromosomes are and what ribosomes and bits and pieces. So it really is a readable book, and it will give you detail to more detail to what we're describing tonight. Do you have any questions before we kick off? Yes. Okay. Probably I'll just get the speakers to speak more closely to the microphone. Yeah. No, that was someone else who's talking about TB, and I'm not sure it was a third, not me, luckily. As long as we all wash our hands a lot and don't get too close to each other, we should be all right. So I'd like to introduce Peter Cullis. Now, what we're doing tonight is, rather than me doing a big introduction, because we really want to get to the meat of this, we're asking the speakers to actually do a short introduction, and then I'm just going to put the timer on, and when it gets to 7 minutes, or about 7 minutes, I'm going to give them the hook. Peter Cullis.
Okay, thanks, Martin. I better speak quickly. This is clear. Luckily, he doesn't really have a hook. Sorry. Just by way of introduction, I'm director of the Life Sciences Institute at UBC. We've also formed an initiative called the Personalized Medicine Initiative that actually almost all the speakers you're going to hear from tonight are involved with. And we're trying to bring molecularly based medicine to British Columbia and to Canada. So we're setting our sights pretty high, and it's a very ambitious program that we're embarking on, and you'll see some indication of that as we go through this talk. Now, I better get onto my slides, of course, and I better get onto them quickly.
Sorry, I didn't do the conflicts of interest. I'll do that.
Okay. So I'm going to talk about what is personalized medicine. And this is actually a very big topic that is very difficult to fit into 7 minutes. I could give a plug for my book here. If you really want to learn, you should buy the book. But anyway, that's probably not quite the correct thing to do. The point being that we're moving from a present practice to a personalized medicine practice in a very, very in a way that you may not be aware of, but this is happening in many jurisdictions worldwide, and the meaning of this is that we're moving from a physiological examination, trial and error therapy, to often ineffective therapy, to technology enabling much more precise molecular level characterization of you as a human being, which leads to much more accurate diagnosis and much more effective therapy. I mean, that's really the gist of what I'm going to say. Now, why do we need personalized medicine? And you all know the answer to this is because you're different from the person next to you. We're all different from each other. And a big example is that different drugs have different effects on different people. For example, some people that take an anti-cancer drug called doxorubicin, which is given to 1 million people every year in North America, will have an adverse drug reaction where their heart will fail. So this is really not a good way to treat their cancer. They'll end up needing a heart transplant and still have cancer. So this is a genetic problem. In other words, if we knew their genetic profile, we could avoid giving them that drug. And so if we sequence our genomes, we're going to find out to a remarkably accurate extent as time moves on who is going to respond well the drug will work, and won't have any toxic side effect. To people the drug will not work on, but they won't have any side effect. Toxic but beneficial in this case, etc. So you better parse out into the populations who will benefit, the populations who won't, the populations who will benefit maybe have some toxicity, etc. Now we all want to know this. 75% of cancer drugs do not work on the person they're given to, and you all know how the nasty effects of these drugs. And so the ability to another thing is that we're not just dealing here with the genomics. There are many other aspects of you that are much more directly useful as diagnostics, and these are the molecules in your blood. And I'm just going to put these down here. Oh, I'm going back. I have to go backwards now. But there's more than 10,000 proteins in your blood, and your blood bathes every organ in your body, and each of these organs actually can leach out a certain subset of proteins. Detecting a subset of those proteins can tell you from a single blood test, or can potentially tell you, we haven't quite got there yet, the health of any organ in your body. And so you can see how we're going to get, in a very accurate way, diagnosis, and also earlier diagnosis, because cancers leach out these similar proteins of their own in a similar way. So you're going to find that the blood is becoming increasingly a window to your health and what you're trending towards. And we're soon going to be able to measure hundreds of proteins in order to enable this. The other category, another category that you should be aware of is all the bugs that live in your body. You contain 10 times as many bacterial cells and related cells in your body as you do the cells that make you up as a person. And this is a symbiotic relationship. And so the interaction between your body and those bacteria really determines how well your immune function works. And so it can lead to many disorders which many of you may be familiar with. So again, characterizing, say, 2,000 or more bacteria in your microbiome, as it's termed, will give you, again, another indication of your health that can be extremely useful. This is just now to sum up. Personalized medicine is really possible because we now have new and relatively inexpensive. They're not that cheap, but they're affordable for many of us. New inexpensive technologies that can be used, the molecules that we're made of, characterizing you right down, you as an individual, right down to the molecular level. This is an amazing advance. I mean, we've never had this possible before in human history. And so it really allows us to be treated as individuals compared to the average. So this is your genome, your proteome, the proteins, for example, in your blood, your metabolome, your metabolites, which reflect the food that you eat, for example. So this will give you guidance as to what food works for you and what doesn't. Microbiome and many other ohms soon to come. So it's going to allow early and precise detection of disease, better matching of treatment to disease, and much more effective preventive care, because we're going to detect disease well before it becomes a problem. I hope I'm finished in something like the right time.
5 minutes, 30 seconds. Not bad. Bruce McManus is promising an amazing set of tests for people with chronic obstructive pulmonary disease. No pressure, Bruce. You have a counter there which actually has your time.
Thank you very much. Thank you, Martin. Can you hear me? It's wonderful to be here this evening with all of you and with Peter. I view these short vignettes as the appetizer. The discussion will be the main course, and the dessert will follow when you have discussions after you leave this room. So my topic, as you can see on the screen, is to follow from Dr. Cullis's introduction. How can we harness knowledge of all of these molecules? And I'm among other things, I lead a center called the Center of Excellence for Prevention of Organ Failure, which really does an analysis of all of the proteins, the RNA, and metabolites that roam around in our bloodstream, and then using computationally driven approach, try to reduce that complexity to signatures that could become blood tests in the clinical laboratory. So this evening, when I talk about markers, these are magic markers in a way. So personal care will rely, and already does to some degree, rely on these magic markers. Of course, they're not the markers that you use to put wow in your presentation. They're not your hair color, your hat size, the shape of your nose, or your smile, even though you can get that information from 23andMe as well. But these are about the DNA, as well as the RNA, the proteins, metabolites in your blood, your urine, your CSF, your cerebral spinal fluid, and other fluids like your joint fluid that can be measured. And they're called biomarkers because they're biological and they're markers of your biology. And then if you say, well, how can these magic markers help? So you get a room full of really interesting-looking people, diverse people like yourselves or like those on the slide. You obtain with consent, ethics-frame consent, precious blood samples, and then you take them to a qualified laboratory and you evaluate those samples thoroughly with tools and technology that can do that, and then you replicate it, and then you validate it, and then you can have a solution that could go truly into clinical care and improve prevention because it can predict the occurrence of disease, it can diagnose it, it can prognose it, or suggest that you might get worse or better, and also indicate whether you might respond to therapy that Dr. Cullis was talking about. And so this is the cycle by which looking at the blood could improve our health. I'll use, as was mentioned by Martin Dawes, two examples in 2 slides. Biomarkers which really can make complexity simple. So chronic obstructive lung disease, or some people know it as emphysema, is a destruction of the lung, often associated with tobacco use, but not always. And it's evaluated often in clinical practice by blowing into a tube and expiring air into the tube in 1 second. This is a test that's not used by many physicians or patients because it's cumbersome and it's very insensitive and nonspecific. But we need a test not only to detect whether COPD is there and how severe it is, but also how active it is, and also a test to predict who's going to have what are called lung attacks, or the acute worsening of COPD that leads to the largest number of patients coming to the emergency rooms in hospitals across Canada of any condition in the country. And so it's a huge cost burden and personally very life-threatening. The same is true for heart failure. If you don't live in Vancouver or proximate to a tertiary care center where you have a qualified echocardiographer to look at the function of your heart, there's only a few molecules, one particularly, BNP, brain natriuretic peptide, that can be used to assess whether you have heart failure. But that molecule and its similar look-alike molecules are very insensitive and very nonspecific in separating those patients who truly have heart failure from those who do not. So we don't have a cheap tool in order to evaluate another very common reason that people show up in hospitals in acute care. So biomarkers can bring simplicity, but one molecule, often we're measuring in laboratories, one molecule to indicate how a system is doing. But if you look at that cross-section of the body there, and you look at how complex humanoids really are, you can see that one molecule is unlikely to expose the complexity of the system and represent it in a precise or accurate way. So let me give you an example. Is this theoretical? No, we measure thousands of RNAs in the blood, like 29,000 of them, 1,000 proteins, and then we have a computation team, a very skilled, internationally leading computation team, and then they, like a magician, like magic markers, they wave their hat, and out of that comes, if you look at heart failure patients, if you see on the slide, in the green, you have a number of genes or RNA that are elevated above the normal range, and you have a number that are below, the same for the proteins. But when patients have their heart failure treated effectively by standard of care therapy, you revert those proteins and genes into the normal range, a significant number of them, that then can become a signature that now is a signature of 17 proteins actually, that is a response to therapy biomarker sets that can tell physicians that their heart failure is being treated appropriately, or that drug works in that group of patients. So this is the direction that it's going. And like Peter had on one of his slides, similarly, this means that doctors who care for patients with heart failure will have new ways to tell whether therapies are working, could be reduced, need to be changed, or need to be increased. And until now, we don't have that kind of guidance in a very precise or specific fashion. So the same is true for COPD. I won't show you much about the example, other than to elaborate to say that lung attacks are the leading cost center in emergency room admissions in Canada and the United States and all other developed countries in the world. And so how can we reduce that? By being able to predict who's susceptible to having a lung attack and preventing them from having the lung attack by more intense therapy and management. And this approach will allow doctors to specifically target patients who need to have more vigorous therapy. It'll also secondarily allow drug companies to develop better therapies to prevent these costly attacks. And we're working on a major project on this specific topic with Genome Canada funding. So in closing, I wanted to bring us back to personalized medicine, or person-specific care is a term I like better. I do not like precision medicine because if you're precise and inaccurate is of no value. It's like a dartboard. And so it's become popular, but it's a very poor term. So person-specific care. We are together going to be able to predict. Knowing you are at risk for diseases allows you to make lifestyle and environmental changes. That's often lost in this conversation, but this is really a troika of environment, behavior, and genes. Prevention, lifestyle, and environmental changes can keep you out of the healthcare system for a longer time. It can be more personal. The right care for you. Doctors need new tools to do that. And then partnership. The real thing that's been missing often in healthcare is, regardless of these new tools, is patients and the public need to be a crucial part of the healthcare team. And then finally, the partnership is what brings us all to full circle to do what we're all interested in, is living healthier, a little bit longer lives. Thank you very much.
What I hope you're getting is some of the excitement. One of the things that I discovered was that genes can be turned on and off by something as simple as activity, which was, for me, a discovery. Ida, do you want to come up and tell us what it was like to do some of this stuff? And I'll let you explain your background that's relevant, because she wears many hats, and I'm not sure which one it's going to be tonight.
Well, thank you very much. My background is quite different from the two individuals that you've just heard from. I'm the former CEO of Vancouver Coastal Health Authority, and I'm currently the vice chair of Genome BC, which is a nonprofit organization focused on research into genomics. So I come at this from the perspective of an administrator. I'm very interested in how the healthcare system works and how healthcare can be improved, how it will change as a result of genomic discoveries. So one of the things that I did, which I'd like to talk about a little bit, was to have my whole genome sequenced last year. There are still a relatively small number of people around the world that have had their whole genome sequenced, certainly many more than 3 or 4 years ago. But it's still an experience which is full of possibility and potential. So let me tell you a little bit about that. Oh, I'm going backwards. So the first question, of course, is why would I do that? I had done the 23andMe program, if you will. Some of you have done that, which gives a bit of a taste of the whole genome. But when I joined the Genome BC board, and I was surrounded by people like Bruce and Peter and many others who were just brimming with excitement and enthusiasm over all the possibilities that the genome, and now, as you heard from them, other omics can represent for individuals. And when I first joined the Genome BC board, the first human genome had been sequenced only a few years before. It cost, to do one, $2.7 billion and took 13 years. So that the possibility that ordinary people like me would actually have their genome sequenced was pretty remote. But then technology took over, and the incredible strides that technology made brought the price down by 2012 to $48,000, which was still a little rich, to 2013 when the price had come down to $5,000. So at that point I thought, well, I think this is something I should do, and the program was offered by an organization called Illumina. They were offering it to groups of 50 people, most of whom were researchers or physicians, and because of my position with Genome BC, I was accepted. So the actual experience is fairly straightforward. A physician takes blood sample or supervises your taking blood samples. Those are sent off to the Illumina lab in California, and you're then provided with both a one-day seminar for information and an iPad with your entire genome on it. So there's my genome. And and of course you start out thinking, well, this is going to be really interesting. I didn't have much history of family disease, so I wasn't particularly worried, and I was really keen. Until the day starts to get closer and closer, and you start to think, well, you know, maybe they are going to come out with something in this information that I'm not going to be that thrilled about. So the actual day when we had our iPads presented to us and the information given to us, it was one of those, okay, well, I wonder what I am going to find out. As it turned out, the Illumina group fully interpreted 1,600 genes associated with 1,200 disease conditions. I had 5,507 variants that they looked at in depth. So that's all sounding a little bit scary. But as it turned out, 0 were pathogenic, 0 were likely pathogenic, and 0 were likely suspicious. So I thought, wow, that's a lot of money and a lot of time and a lot of effort to come back and tell me that there's kind of 0 that these folks have found. Fortunately, Brad Popovich, who's the Chief Scientific Officer at Genome BC, was also taking the same program. And he was so excited because he said, here's your entire genome. Every new gene that's discovered in terms of having significance, I just have to go into my little iPad and I can look it up and see whether I have a variant. And of course, as many of you know, genes can tell you a lot about disease states, but they also can do other interesting things, like, for example, aging, hair color, intelligence, and so forth and so forth. So I've looked this up now several times, including recently when my nephew, who is a year old, potentially had a genetic disease. And although it was a nephew, I could still go in here and see if I had the genetic variant that was potentially implicated. So it's a very, very exciting opportunity. I learned some other things. The first and probably the most major thing, if you will, was it's very complicated. There are 6 billion, and I've already been told that's actually not base pairs, it's actually building blocks in your DNA, but it's a big number. I had 3 million variants, and although the sequencing is now down to about $1,000, the interpretation is still very expensive. So to interpret my genome took a team of about 6 or 7 people who spent 30 hours on each of the participants, making sure they had done the interpretation correctly. So the big issue right now facing scientists and individuals is how do we get more information into the research system so that you can identify all these genes which are being collected at $1,000 a pop. It's not that difficult to collect them. But unless they're correlated with a health condition, they don't actually mean very much. For example, APOE gene didn't mean very much until there was a link with Alzheimer's. So what we need today is to have more and more people put their genetic information along with their health information into research and into databases. And it's moving forward. The UK has introduced a program where they are analyzing 100,000 genomes, so people are giving their health information and their genomes. President Obama just announced the U.S. will look for 1 million genetic volunteers. Give us your genes and give us your health information. And then all those variants, the 3 million variants I have, will start to mean more than they currently are. The other thing that I wanted to mention is I think there are opportunities for BC where all of us have most of our health data with the province. I've given permission to use my health data to several companies and organizations, research groups, all in the U.S. Why? Because nobody in Canada has asked me for it. No one in Canada would know what to do with it at the moment. So there's an opportunity here that all of us, I think, within this province can think about. How can we start to take advantage and participate into the research that's going on? The other thing that I learned, oh, and by the way, this is what it looks like, which is, as I say, complicated. So I'm not going to go through all that. I didn't understand most of it. They explained it all to me, most of which I've now forgotten. So what we need, obviously, is simplification. But the other thing that I've learned is part of the huge change that's going to go on with people who have their genome is it's now mine. It's mine. So what's the role of my physician when I've got the data and I can look it up? Some of you have done the 23andMe experience, where you can go into your data and you can not only get information about what genes you have or what you have, what you're at risk for, but they tell you all kinds of other information as well. They allow or encourage you to bring your family data. So my sister and I can compare notes. So I am at higher risk for gallbladder disease relative to the rest of the population. She's at higher risk for coronary heart disease. Both of them are in my mother's family, so we know there's a genetic link. So there's a lot going on right now, but the opportunity that's in front of us is how can we get the physicians in the healthcare system to collaborate with individuals who have all this data so that it's not I'm here with my data, what am I going to do with it? And physicians saying, well, I'm not sure what your data means and I'm not really interested in using it, which we know we have. So I think huge opportunities for transformation.
Thank you very much indeed. If your brains feel tired, now is the moment we can have relief because we're going to find a way of preventing brain failure. Isn't that right, Teresa?
Yeah, absolutely.
Great. And so you will have, your time is there. But
perfect. Just going to bring the microphone a little bit. Can everyone hear me now? Okay, great. So just a very brief introduction to me. So before I went into research, so I'm an associate professor at the University of British Columbia. I was a physical therapist. Practiced for many years, worked within the realm of sports medicine, was involved in sports all my life. So I suppose it's not too surprising that right now in terms of my research program, I focus primarily on healthy aging. And so what I'd like to do in the next 5 minutes is really shift the focus away from the world of genomics and molecules. Do I get to still stay? I'm not going to get kicked out? Okay, perfect. And shift the focus more towards prevention, specifically with the lens of how you could potentially modify your own risk for certain diseases through what I would consider as rather global strategies. So the question that I'm asked to really address is brain failure inevitable? I have 5 minutes, so the short answer is no. And just a brief explanation. So, I mean, we all recognize that as you age, there are changes in your cognition, sometimes mild, sometimes more severe. And we also know there's actually physical changes that do occur in your brain as you age. However, we also, I think, through observation and just interacting with individuals on an everyday basis, we realize everyone ages differently, right? Both physically and cognitively. So I'm sure many of you can think of at least one person, whether it's a colleague, a friend, a family member who's aged really well. They're still really witty in their old age, just as witty as they were in their younger years, right? So in terms of, a popular icon, it would be George Burns. He managed to, entertain all of us with his quick wit well into his late years. So there is a lot of variability. And conversely, I'm sure you can think of someone who didn't age very well, maybe in their 50s or 60s, they're already showing some significant deficits. Now, for those who do age well cognitively, I can imagine many of you think, well, they were born with a set of really good genetics. They're just born healthy. They have good set of, profile that they really don't have a risk for developing dementia or associated diseases. However, that's not necessarily true. That might be true for some individuals, but we actually know that there's a good number of individuals who do age very well, but if you actually put them under assessment, so if we scan their brains, we can actually see very frank evidence of brain disease. Yet, behaviorally, they're doing really, really well. So why is that? So what contributes to this sort of variability we observe in everyday life? Well, first of all, it is genetics. It does determine some outcomes, but it can be modified. So Dr. Dawes did allude to the fact that your environment can interact with your genes and modify the expression of those genetic predispositions. So what modifies your genetics is your environment. So in terms of environment, as it relates to your cognitive health, we're talking about your level of education or a level of intellectual engagement throughout your lifespan. Okay, physical activity is key, nutrition, your degree of social network, and as well as the quality and quantity of your sleep. All these three components really contribute to what we call reserve, or if you can imagine, your RSP. So your buffer towards aging. Okay, so if you imagine if you did a really good financial planning, you started early, you invested, you followed your advisor's advice, hopefully a good advisor, you have a lot more sort of, degree of safety towards any financial downfalls or unexpected expenses that you may incur in your later years. This is the same thing with your brain. You can build it up throughout in your entire lifespan. Okay, and when we talk about reserve, there's two concepts. One is brain reserve, and that really relates to how much brains do you have? Okay, how big is your brain, and how well connected are the different regions of your brain? Physically, how well are they connected? The second component is really, really interesting from the concept of neuroscience, and that's actually relating to how well your brain can adapt functionally. So that's called cognitive reserve. So if you think of brain reserve as sort of the hardware of a computer system and cognitive reserve as your software, okay? And combined, that really determines how well you can, again, adapt to both disease and also even to trauma. Let's say if today you suffered a stroke or even had a traumatic brain injury. If you have greater brain reserve to start off with, your outcomes might be actually better. And then moving forward for the next few minutes, I'm just going to really highlight the role of this activity, okay? And why? Well, let's start off with this question. Again, RSP season. You go into your advisor's office and they promise you, or he or she promises you, an investment that will give you a return of 100 to 200%. Would you take it? Anyone wouldn't? Okay. Well, that's kind of the evidence we're seeing for physical activity, believe it or not. So a study in 2011 actually showed if you simply walked at a moderate pace for 3 times per week, so up to 120 minutes, you can actually reverse your brain age by up to 2 years. Okay, as you age, again, on average, your brain does shrink, and certain structures shrink more than other regions. Okay, and here in this study, they actually found that the memory center actually not only maintained volume, but actually grew. All right, so it was actually literally reversing your brain age. Second fact, the observed benefit was also noted among individuals who we know are at risk for dementia, but only took 6 months of training and twice a week. That's not what you would have expected, would you? But we're actually seeing that it seems like those individuals who are at actually greater risk seem to benefit more so from physical activity. And then, third fact, if you don't like walking, running, swimming, anything that increases your heart rate, then lift weights. So we're actually now demonstrating that a broad spectrum of physical activity are beneficial for your brain. So it doesn't always have to be aerobic activity, it could be other forms of activity. And then when we talk about dementia, drugs always appear on the forefront. what is the latest drug, and what's its effectiveness. The reality is we currently do not have an effective pharmaceutical strategy towards treating dementia. And based on the current evidence, the magnitude of benefit we have observed across all the different exercise trials exceed any study that's been done today on the drug trial. And then the last piece is that it's also been estimated if we could just reduce physical inactivity by 25%, we could actually prevent 1 million cases of dementia worldwide. So pretty powerful. Again, I'm sure this is a message all of you heard, but what I'm here to do today is really try to emphasize the potency of physical activity. Sorry, I'm having a hard time speaking, seems like. Okay, so how does physical activity do it? Well, first of all, I don't think we should be surprised because the brain is all of 3 pounds, but it actually consumes 20% of your body's supplies, blood and oxygen. It's literally your lung and heart's best customer, right? So reason would indicate that, you know, a physically healthy body would house a physically healthy mind, right? So by being physically healthy, you're always maintaining the necessary blood and oxygen and nutrients to your brain. Again, the brain is highly, highly metabolic. That's why it requires so much blood and oxygen at any given time. Being physically active also reduces your key risk factors for developing chronic conditions that we know are highly associated with dementia, such as diabetes and high blood pressure or high cholesterol. And the third piece is that anyone in this room taking vitamins in the hopes of promoting the brain health, whether it's omega, anyone? Okay. Did you know when you exercise, that act alone produces what I call brain vitamins? So when you exercise, it actually increases what we call growth factors in the brain. Growth factors actually promote neurogenesis and survival. And when you exercise, whether it's aerobically or strength training, you actually can increase the level of circulating, okay? And thereby, it actually promotes brain health very much in a direct way, not just through by promoting physical health. And we actually do know that different types of exercise may promote different types of growth factors. So for resistance training, we tend to promote the growth factor of IGF-1, and aerobic training may promote something called BDNF. Okay, and I won't go into it now. We can maybe discuss later, but this is where we can personalize some of the training based on your genetics. And the last piece is the good news. To get the benefit of this activity, you don't need to qualify for the Boston Marathon. And why? And the bad news is obviously you're running out of excuses to be physically active. So facts, starting in late life is not futile. Evidence would suggest that even if you start today, you would still reap the benefits for your brain. 2, even walking just 1 kilometer per day can protect your brain. So across a 10-year study, those who walk 1 kilometer on a consistent basis at baseline actually maintained their brain volume 10 years later and also had reduced risk of dementia. And for those of you who, again, have a hard time getting started, this is what 1 kilometer looks like. I won't go into the details, but if you look at the time and the distances, very, very manageable. And again, if I still haven't convinced some of you, I'm going to have a lowball challenge to all of you now, which is limit your sitting or lying time just down to 23.5 hours. Okay? That's it.
Thanks very much, Teresa.
Okay, on to you, Larry. Ready for the last stretch? Just 7 more minutes, and then I'll get you doing some activity just to preserve your brain. Larry, okay.
Thank you, Martin. Good evening, everyone. Just by way of a quick introduction, similar to Teresa, in my first life I was a pharmacist. I worked in practice. I practiced for 10 years, so I've been involved in the healthcare system. I have an idea how it works. I've moved on. Crossed over to the dark side, moved into academia. Now I'm an epidemiologist, health economist, and my life is around taking what these guys develop and trying to figure out if it has any effect on us. Does it improve our outcomes? Does it improve our quality of life? Does it reduce hospitalizations? And how much does it cost us? I'll also reveal my bias as I am a patient. I don't know how many of you are patients, but I am a patient. I've had my vaccination this year, so by default I am a patient. I think we're all patients. I don't have any chronic diseases yet, touch wood, but I have a vested bias and a vested interest in
hoping that the healthcare system will be there to take care of me when I need it. So I'm going to take a little bit different flavor to what we've been talking about. And Peter alluded to this. In the healthcare system, we really do treat everybody as a one-size-fits-all. Everybody's kind of an average patient. We have our randomized controlled trials. In our trials, some people respond, some people don't, but on average they do. So we take our drugs and we will treat the next patient that we see that on average we expect that they're going to benefit, but we're just not quite sure. We have to wait and see. I'll give you an example. Who knows how much our provincial budget is that we spend on drugs every year in British Columbia? Any idea? Okay, $1 billion. It was $1.1 billion. It was just reduced by 10% last year. So now we spend $1 billion. We spend 10% of our $1 billion budget on 5 drugs. They're biologics, which we use in rheumatoid arthritis, ulcerative colitis, psoriatic arthritis. So we spend $1 billion on those drugs. In rheumatoid arthritis, they cost about $20,000 per year per patient. They work really well. They are awesome drugs for the patients that they work in. Unfortunately, for example, in rheumatoid arthritis, only about 60% of patients respond to the first drug that we put them on, and it takes us 6 months to figure out if they're going to respond. So it costs us $10,000 worth of drug before we can actually figure out if a patient's going to respond, and 40% don't respond, so we've just thrown away $10,000 worth of drug on 40 patients. Then we move them to the next drug. They might not respond to that one either. So Peter alluded to finding patients who have, adverse drug reactions, but, there's significant potential benefit to be able to find patients who are going to respond when we treat them initially so we don't waste all this drug and money. So treating people more individually. So here I didn't actually hear, nobody talked too much about cost, so we're just going to put a different lens on things and look at the cost. So here's the latest data of what we spend in Canada on health care. In 2014 was $215 billion. If you'll allow me, I'll not talk about health care and we'll talk about sick care. Because we actually try and rescue people once they've developed diseases and they get sick. So this is what we're spending on sick care, right, once we get there. So about 2.1% growth, about $6,000 per person. The younger in the crowd, they don't cost us very much. I'm kind of at the front end of that curve where we're starting to go up. I can tell you I'm just probably encroaching past around the average 6,000, and as we get into our 70s and 80s, I can tell you we're significantly higher than $6,000. And we spend about 11% of our GDP. Here's how much we spend in our provinces. You can see BC here, if you can see that. On average in BC, we spend about $5,800 in British Columbia per person on sick care, which is about 43% of our annual provincial budget. So if you allow me, let me just round up and we'll say half. So right now we spend half of our provincial budget on sick care. The other half gets to go to environment and education, transportation, social programs, putting roofs on BC Place. I mean, this is all part of it, right? But we spend 50% on sick care, and the other 50% we have to manage with everything else. Not quite 50%. We're 43%. Newfoundland is almost 50%. They were 50% the year before. I think they're down a little bit to 47. So just keep that in mind that we're already spending half of the taxes that we pay on sick care. We talk about this in health economics is we unfortunately we have this culture. It's new. I want it. What is it? New always sounds awesome. It's new. It's got to be awesome. It's got to be fantastic. I want whatever's new. We're finding more and more, though, that lots of our new technologies and the things that we can do aren't necessarily providing us the benefit at the patient level that we would expect. Notwithstanding, there are certainly lots that do, but they all don't, and that's the question. So here's the problem. Our options and our programs that we can invest in are almost unlimited. Again, I'll reveal my bias. I'm kind of bringing a different slant to this, but I think this whole personalized medicine approach is actually really exciting. Peter said, can we do it? And the key is that we can. The question is what we can do, what should we do? What should we adopt, and what are the benefits to some of those pieces that we can adopt at the end of the day? So our options are almost endless. Our resources are finite. We know we spend 50% of our budget on health care or sick care at this point. The only thing that we can do if we want to add new things is start to cut things. We can cut things in health care if we want to add new things to health care. We can cut more teachers. We can not put SkyTrains in. We can cut other places, but if the pot doesn't get any bigger, we have to cut if we're going to add on. Or we can do a referendum and see if maybe you all want to spend another 1% sales tax to add more to the health care budget, the same as we're doing for our transit. Same thing. We either have to make the pot bigger or disinvest. But in the end of the day, choices have to be made. These are the difficult choices, right? Where do we invest? Where are we going to get value for money? And where are we going to get return on investment for our healthcare dollar? So the question is, and this has been brought up a few times, are we at a stage where we're at a paradigm shift, where maybe we can be moving more to healthcare and less to sick care, and in fact the value at the end of the day is going to be better to society? I know we're all affected by our health. Having a good healthcare system is really, really important to us, and I would absolutely agree. I'm part of it. We're all part of it. But the question is, can we invest our money in different ways that we can? And I just wanted to wrap up by using this analogy that a colleague of mine used the other day. So if you can see, we got a guy hanging from a rope here. We got this bridge that really didn't work. So if you think of the river as being our healthcare system, and once you fall in the healthcare system, we spend a lot of time getting people out of the river. Here we've managed. We've got a bit of a bridge here, and we've got a way to maybe keep people out of the river. But maybe if we actually built a better bridge and kept people from falling in the river, it actually might be a more efficient approach than trying to fish people out of the river all the time. So that might be just an option and just a way that we could look at how we might allocate our finite resources at a societal level to actually for the benefit of all of us. And that's the million-dollar question.
Thanks. The reason I came to BC, because you can tell from my accent I didn't actually grow up here, was the amazing collaboration that you see. And what you've had demonstrated here is people working together but coming from very different angles to look at the same problem with different lenses. That's what makes BC different. And probably gives us the lowest cost per patient we look after in the illness sector. But it's an exciting group to work with, very exciting for me as a family physician. And I hope you've felt some of that excitement as coming across from the speakers as they've talked through. Now, what I'd like you to do is we'll take 5 minutes, so just stand up. We're going to extend your life by about 30 seconds and just move around a little bit, but stay around your tables, and we're going to come around and collect your pieces of paper, and then as a group we're going to quickly start looking through them and start answering some of your questions. Okay with all that? Good. Okay, let's stand up. Need it. So thank you for an incredible list of questions. I'm just going to take one that's addressed the insurance question, because will doing genetic tests affect my insurance is the gist of it. And I went to a talk by one of the directors of Standard Life, so I think they deal with about 10 million Canadians, and they said, absolutely not. It makes no difference. They've had actuarial data that says our average age is, our average lifespan, is going to be 78. And knowing a few people have a predisposition to genetic disabilities and may shorten their life makes no difference to the overall payment that 10 million people will pay. And it would be too much work for them to sort of quantify for each individual based on their genetics to say, okay, you need to pay 500 because you've got something that might increase your risk by 10%. They said it just is not feasible for them to do. So at the moment, that was quite reassuring that we could get the genetic test and it's not going to put our premiums up to a ridiculous amount. The other thing is we live in Canada. So, healthcare, once you're ill, is there ready for you. It might be different south of the border. But it was really interesting because I came into the audience thinking, okay, if I have my full genome done, then the insurance companies are going to take it and get very interested. They're not interested. They know from family history whether you have Huntington's chorea. That's out there. They know about other family history. They ask that already. So the additional benefit to them for doing genetic tests or to be able to quantify your risk exactly, it's just not there. So we had some interesting questions, and I thought we ought to start off with one from to Teresa about the gender issue. Teresa?
So just to share the question to begin with, the question is, do men's brains shrink faster than women's brains? My life could be on the line here.
It's being recorded.
Actually, the reality is, based on what we do know, no. If anything, women seem to be more prone to atrophy with age, and as well seems to be more prone to cognitive decline. Why that is, it may be related to sex hormones, especially estrogen. So the female brain is very sensitive to estrogen, and there's different types of estrogen, and certainly with major hormonal events such as menopause, there is an effect on the brain with certainty. So I'm not sure who wrote this and how he or she was hoping I would answer, but I would say no.
So the only thing, Teresa, that isn't in that question is, or in the answer is that men start out with a smaller brain, though. And you know that whoever wrote it was thinking that.
Yeah, Bruce could have been really quite clever if he had come out as a woman.
Touché. You don't know.
Bruce, would you like to deal, as you've got the microphone, with one of your questions?
After that introduction, yes. I think what I'll do is there's a question that's two parts, and I'll draw Peter and Martin in on this as well. The question is, what type of omics are most advanced currently, and I'll give you the second part too. What type of omics can BC lead in using in practice? And so this would be, as I like to say, a two-beer conversation, because it's not a simple answer. However, in terms of in relationship to different illnesses, let's say in oncology, or in, as I alluded to, in something like heart failure, the approaches and the utilities of some of these for complex chronic diseases like heart failure that may not have an obvious genetic defect, a mutation, a deletion, or the like. The approach may be something like I described, where looking at the tens of thousands of analytes in the blood and then computationally reducing them into a signature can be very useful, and we're right there already. On the other hand, in oncology, there's already very good demonstration. In fact, that's sort of been the testing ground for showing that personalized approach can work, where people respond to therapies, for example, to receptors in certain breast cancer populations, for example. So there it would be more of a genetic approach, which is also called omics, but it's DNA, whereas in the blood it would be RNA and proteins and metabolites. So I would say that the utilities are across the board, and they're different for different disease states at different points in your life cycle. And then I'd finish by passing it back to Martin, because I think that you should mention the project that you're leading that is a very important project under the Personalized Medicine Initiative in British Columbia that could be a leadership position. So why don't you...
Okay. Well, so before I do that, I tend to divide genetics into three areas. Those tests that will help me predict, first of all, how a disease is going to go forward, or whether someone will get a disease. Second thing is in helping me make a diagnosis of a patient. And the third thing is treating a patient. And that's where we're focusing in primary care. Because you've heard, we don't know whether you're going to gain benefit or harm from the drug I'm about to prescribe you. And trial and error is no longer the way we should do it. We actually have a collection of tests that we can do quite cheaply that will help us predict whether you'll have adverse events from a simple drug like codeine, which if you ultra-metabolize switches into morphine really, really fast, and it can suppress your respiration and actually cause you significant harm and sometimes death. And you can't predict that without doing the genetic test. The genetic test is very simple. So what we're doing is saying, let's not do one test for one drug. Let's take 26 diseases, lump all the tests that we can do for the drugs used in those 26 diseases, and then test that in primary care. That's the project we're doing. 250 patients will be recruited in the early summer, and then we'll move to 6,000 patients in the fall, having developed the mechanisms to integrate all this information into electronic medical records so that if I put in the disease asthma, it takes everything into account and comes up with the recommendations, including any genetic abnormalities and predictions for which drugs should be used. So that's what we hope to be announcing publicly, so it shouldn't have done it tonight, in about 3 months' time. Peter, you've got a question.
The microphone in this direction. So a question I have is how accessible are these tests for biomarkers? In other words, how accessible are these tests for your genomics, the proteins in your blood that are diagnostic, the metabolites, as well as all the bugs that live in you and on you? And the answer is, they are accessible, but they're not accessible through the normal healthcare system. There are now, we'll see how this unfolds over the next few years. And so you heard how Ida had her genome sequence, but that's now a $5,000 investment. That will tell you something about the risks of your diseases, or risks of disease. There are companies springing up throughout North America and Europe that will now, for $100, or the price varies, will give you a measure of 100 or 200 proteins in your blood, or metabolites, or 2,000 bacteria in your colon. The trouble is that, as Bruce was saying, that often that information is not all that useful to you because it's hard to interpret it in terms of what you've actually got. And so that's where the population-wide efforts, as we get this data in from more and more people suffering from various conditions, is going to become much more definitive. So when you get these tests, you'll then be able to go do the analysis, the bioinformatics as it's termed, to get real information that pertains to you. So this is going to be a process that evolves over the next few years, but I think in 5 years' time we're going to start to see a real swing towards this process that gives you much more detailed and accurate diagnostic information about you. So,
thank you. Larry, do you want to take up one of yours?
Okay, thank you, Martin. I can probably kill 2 birds with 1 stone. I've got 2 questions here, and the first one was a question around, and probably just stemmed from the fact that I went through one of my slides very quickly, and there was questions around how does BC measure up against the other provinces for what we invest in our healthcare system. So, Martin, if you just wanted to go back to that slide, it's just a couple back. We could leave that up for a minute, and a few people might want to. Yeah, no, next one. That one. So hopefully you can see that, and that will sort of give you an idea of where BC measures up, and maybe that'll influence whether you vote Liberal or NDP or what you might do next time. I don't know, but anyways, and that might stimulate some more questions. The other question was, any advice for students to get into the field of personalized medicine? And I would say, well, it depends. It depends what you want to do, and I think, Peter and Bruce and I obviously talk very different languages, but we're working as a means to the same end. So, I sounded a little bit like a pundit, I think, up there, that I was maybe a little bit negative, but in fact, really what we're trying to do is they're doing the work on finding the really good biomarkers or the really good omic tests that we can help identify some of these things that might actually influence outcomes for the patients at the end of the day. So when things get down to my end of the room, we can do some evaluation to say, okay, when we go to the payers, when we go to the Minister of Health and say, this is something that we should be integrating into our health care system. Is it cost effective? Does it improve the health outcomes of our patients, or does it potentially reduce costs? So we can look at Does it offset the costs of physician visits, of hospitalizations, of adverse drug reactions, and is this an efficient use of resources? So if you are a student looking to get into personalized medicine, you can run the gambit right from the basic science end right up to, I can tell you that that within the ministry, they're having difficulties even dealing with the policy end, given that we have these new genetic tests for drugs that might actually that might predict whether you respond or don't respond. The unfortunate part is the tests are out of one budget and the drugs are out of the other budget, but you can't get the drug without the test. So they're not quite sure how to figure out how to pay for these things. Is who's going to pay for the test when somebody else has to pay for the drugs?
You mean healthcare isn't one budget?
If only it was that simple. So I hope that answered your question.
So Larry, I just wanted to add one or two things to the comment about mentoring and training for student opportunities. And it alludes to one of the comments that either Martin or Peter made earlier about BC being a highly collaborative health and health research environment. But if you really are interested, you need to work with a number of the organizations that we all lead. There's probably 10 or 15 of these that are networked together in BC, where people are trained deeply, and the mentors are trained deeply but think broadly. And so they reach across all of the disciplinary boundaries, and they think from molecule to population, and they think from mechanisms to health economics. So all of the programs that we have all elements of that in it, and you can go in those environments, and then you can gravitate in one direction or the other and get the depth that you want. But there's enormous opportunities in British Columbia.
Thank you. Ada.
Thank you. I have several questions here that pertain primarily to 23andMe or direct-to-consumer companies who offer information on genomic tests, not the whole genome. And the questions really refer to how does a member of the public know that the tests are, or the information, the results are clinically valid? Are they complete? Is it appropriate that these companies suggest after people see the results that they should go and talk to their doctor? So I'm going to what I'd like to do, if I could, is just talk a little bit about 23andMe, which, as many of you know, is somewhat controversial. When I went through the experience, 23andMe was still providing health information. And the reason I mention it, the organization, is that the 23andMe offers people a lot of information that otherwise they would only be able to get through tests that their doctor had done. Now, I think that the clinical validity of the tests is fairly robust and good quality. It's done by Illumina and by LabCorp, two very, very reputable organizations. The data that they provide is based on different types of research studies. So if Bruce did a particular study, they might draw on that. If other researchers do tests or provide discovery type of reports, they'll draw on that. So one example, let me give you an example. My highest risk factor using the 23andMe information was for gallstone disease. The average risk for an individual is 14%. For me, it was 22%, which means 22 women out of every 100 with my genotype would develop gallstones. 23andMe tells you what the gene that is involved is. It explains how that gene works. It encodes a cholesterol transporter protein. It goes on to say that the inheritability of gallstones is 25 to 29%. So 70% of the cause of gallstones would be not hereditary, it would be environmental. And then it goes on and talks a bit about what can you do, and then it gives references to all the studies that they're basing those comments on. So for me, 23andMe was a huge educational experience. It was a way to learn a lot more about the research that's going on, and to understand what these gene markers and the alphabet letters, the A's, the C's, the T's, and the G's. So I had for example, with gallbladder, I had a C and a G on that particular gene. My sister had 2 G's. She wasn't at more risk. I was. So just that one indicator changed the risk that I was at. So my experience with it would be it's a way to give people information. If you use it that way, then I think it's a very valid service to be provided. It doesn't take the place of the doctor, nor should it, and I don't think it intends to.
Thank you. Peter, do you want to?
So I have a very personal question. The question is, I am starting chemotherapy soon for cancer, and how can I access genomic testing to see which drugs will be the most effective for me? And one of the answers here, of course, is to be part of the programs that are going on at the BC Cancer Agency, because they have one of the most advanced programs in the world for doing just this, of determining the genetic makeup of the individual's cancer. Now, there are a number of other companies, commercial enterprises, that will also do this. For that, of course, you're going to have to that costs. But this is really something that we're at the start of now, and I think not too much. It's one of these things. One of the things about personalized medicine is that this is going to be driven very much by you, the consumer. As you see the potential power that comes from really knowing in intimate detail the makeup of your body, there's going to be an increasing demand by the population, by individuals, for particular resources. And so that we see this as we're going to be moving away from a more passive recipient of healthcare to a much more informed population. It's going to cause all sorts of strains and stresses on the medical system. I think that's quite clear. But the overall thrust towards understanding yourself and your disease at the molecular level is something that is increasingly going to become vital.
So another reason I came to BC, apart from the beauty and the rain, and was the BC Cancer Agency. It's an amazing organization that's been going one of the longest in Canada, just taking every person who gets cancer, nearly every person who gets cancer in the province, and then following up what goes on with those patients, looking at the therapies, looking at the outcomes, and then revising the therapies and advising whether you live in Fort St. John or whether you live in Trail, you get the same level of care as if you're living right next to the VGH. It's an amazing system. But the added bonus of that system is the tracking of patients to see how they do. And Peter alluded to it a little bit. What we're just starting to do now with the genetics is to track. So the encouragement is for more people to become part of a population who have whole genome done, and then follow over time to see the impacts of medication, of activity, of other lifestyle changes may have on disease occurrence. And I think we, having had the principle set that anyone who gets cancer is automatically linked to the BC Cancer Agency, it should be that anyone who has a genetic test gets it automatically linked and then commits to have their medical information anonymously, it doesn't have to be identified, but followed up over time so that we can actually learn from what we're doing. So, and I think we are well positioned to do that, and we're just talking with the government about how that might happen here. Bruce.
Martin, I might tell a little story, and maybe it brings the stark reality of our opportunity more clearly into focus. I had this in slides I originally prepared, but I eliminated it because it's sort of personal also. But about 5 or 6 years ago, I was speaking in China, and I don't know if the postdoc intended to poison me, but I don't think so. He took me to dinner with his family, a former postdoc, and I had a great meal, except that I think the chicken was bad, and this is an aside. So my wife and I came back back on a plane through San Francisco to Denver, where her brother lives, and I got deathly ill with gastroenteritis, the sickest I've ever been. I was prostrate and in bed for 3 days, and I woke up about 5 days later with a friction rub, which is a little noisy rub that comes from having inflammation in your pleura. And I thought, well, I probably had pneumonia.
Sorry, pleura is the lining of the lung.
Pleura is the smooth surface on the outside of your lung, but it was like gravel, and so I thought I had pneumonia, and as any good doctor, I refused to go to see a doctor. So that's first advice, but that's the first parable. But then I had lunch with a friend on the Sunday. This was on the Saturday, and Sunday had come up from San Francisco about our biomarker work, and I couldn't stay awake and I told him I just had to go home. Well, I was supposed to travel again the next morning, and my wife canceled the flight. I had to go to see my family doc, and then immediately sent me urgently to a respirologist and had a CT scan, and I had a massive pulmonary embolism, which is a blood clot to the lung. And I had no risk factors. I'm healthy, I'm pretty active, and so on. So they did, among other things, I ended up on warfarin for 6 months and so forth. But they did, there currently had become known that there were gene sequence variations, one of them called factor V Leiden, because it was discovered in Leiden. But it's a variation in one of the clotting proteins called factor V, and that it increases all of the things being equal, it increases your risk of having a blood clot, a deep vein thrombosis, and a pulmonary embolism by about 5, 6, 7%. And so it happens so that was interesting. But even as an educated person, I knew that it probably had been at play for me on a long flight, because you get stasis in your veins, and then you have this little extra risk. And then I had dehydration because of my gastroenteritis, and you could think of all of this collective. But I never really thought much of it in my family, even though it's inherited. It's often an autosomal dominant. And so then, about 2.5 years later, one of my 34-year-old nieces, who just delivered her third healthy child, she's an intercollegiate athlete, a very talented person, had calf pain on a Friday night, 2 days after birth, got home, had calf pain, and the next morning she dropped dead with a pulmonary embolism. And so I'm not trying to make you sad, but I'm going to tell the story because it's why you need to know. So people say, well, I don't really know this stuff is interesting, but it's not that relevant. Well, so then, so it turned out I finally woke up, and I have a big family in Western Canada. There's about 100 McManuses. God bless you. But there's a lot of us. And a lot of nieces, nephews, and great nieces and nephews. So I finally got an email loop with the whole family and got, especially the women, to get tested because of pregnancy in young women being a risk factor for DVT anyway, regardless of whether your genes are abnormal. And we got, in the meantime, so this is how behavior, environment, and genes interact. In the meantime, one of my great nieces at the University of Saskatchewan started having minor pulmonary emboli, and she, among about 8 or 10 others out of the family, are positive for this gene sequence variation. But in addition, she was on a very notorious birth control pill called Yaz or Yasmin. As you know, there's big class action suits about this, because what does it do? It causes increased risk of pulmonary embolism and gallbladder disease at a very premature age in young women using this pill. So so I asked her a very, it's a personal question to a young woman, her great uncle or whatever, are you on birth control? And sure enough she was on that drug, plus had the factor V Leiden slight increase, and that's you've got a composite. So the family has recovered from Krista's death, but the profound effect of just knowing that, but not knowing it soon enough. Like I gave the toast to the bride at her wedding, and 10 years later gave the eulogy. So and if I'd have known, and I would have had them all tested beforehand when she had calf pain on Friday night, I could have prevented it. Now I know that this sounds like a very melodramatic story, but in fact it's real, and it sort of shows you, like, just knowing these things interacting. That's why it is environment, behavior, and it is genes, and you just don't know. So it's worth knowing. That's all. Sorry.
So we are talking about health. So we do have stories that don't always have happy endings, and that's why we are passionate about this, because we see it making a difference. Whether we're basic scientists or family physicians, it's relevant to us, and it's relevant to our families. So I'm going to go on to another question, which is sort of going to be about the cost. For an average individual, do you foresee any value add to do whole genome sequencing versus doing sequencing on a small number of genes that have a strong association with diseases of particular interest? And in a way, it's quite a nice question because I don't have to answer it. Why I'm saying that is you saw the price of the testing coming down to $1,000. If you actually go and get the whole Illumina package, it's $5,000 at the moment. But to do the physical test costs $1,000 for your full genome. That will drop again, and by the time we're doing this talk in 3 years' time, it'll probably be around the $200 or $300 mark. And there won't be any point in doing little bits of gene when you can do the whole thing for $200 to $300. Not everyone will getting it for $200 to $300. I mean, that's still a bit of a barrier. But if you need a genetic test, rather than saying we're just going to do this discrete piece of genetic material, we're going to probably try and do the whole thing. Then storing it is going to be an issue. But that's someone else's problem. We have people working on that right here in BC, looking at big data. So the simple answer to that is, at the moment, you will probably have discrete tests because they are under $100 or around $100. You can have a bit of your gene, an exome, done for $500. But we will probably, as I say, do the whole thing in 3 years' time. Larry, you had a question, Adam.
Yeah, so, and maybe just to build on that, Martin, I think there was a question around the value of having your whole genome sequenced, and I think Ida touched on that as well, and you might want to touch on this too, Ida, is I think there is some value, and you spoke about there being some value, but the real value is going to be as we develop the big data. And Ida touched on it. The more people that have their genome sequenced, the more big data that we generate, the more associations we can identify, the better the data that we have to actually help inform the decision. So I think, today there is some value to it, and I think Bruce just alluded to one of the very, distinct points, but one can only assume as we get more data and can investigate those things that the value is just going to go up because we're going to know more and more about those things. There was a question around the average cost of health care per person at $6,000 per patient, and how is the cost split between doctors, drugs, hospitals, and research? Well, I can tell you within that budget there's nothing on research. Within the healthcare budget, we've had this discussion with the Ministry of Health a lot, and they don't pay for research, so there's no research. Drugs account for about 17% of the budget, and the cost of drugs is divided about 2/3 out of the public purse and 1/3 out of the private purse, and about 60% of British Columbians are covered by private health care. The drugs have just recently surpassed doctors, but they're right around the 16 to 17%. Hospitals are up around 1/3 of the budget, so they're certainly the most expensive, and that's hospitals in general. So you have to remember that within the hospital budget are a lot of our really expensive drugs that don't get captured in the drug budget. So slight underestimate of how much we actually spend on drugs and doctors, because they're also embedded in the hospital budget, which is about 1/3. So if you add drugs and doctors and hospitals, we're about 60%, and then the other 40% is, and I was being a little bit facetious when I called it a sick care system, because we do invest in public health, in vaccination programs, and all that other piece of the budget. And so the bottom line question was, how will personalized health care lower the cost? And I think that's the million-dollar question, and I think there are lots of opportunities where it can, and that's really what we're trying to find is what are those key things that we can do, that we should do, that will really actually help to lower the cost, so that in fact we don't necessarily have to disinvest. We don't have to get everybody to pay more taxes so we can do more. Are there real key pieces that we can do that will actually lower the cost, that then will really kind of increase the size of that pot, that we can do more things?
But I think you sell, Larry, yourself short in the sense that I just want to emphasize this in terms of the multidisciplinarity for all of this personalized medicine train going forward. People involved in detailed health economics modeling are engaged in all of the projects that are undertaken. So in the modeling, we know, for example, a blood test to exclude the possibility of acute rejection of a heart in a heart transplant patient will reduce the costs of care for individual patients by over 80%. And beyond the fact that it'll mean that they could have their blood drawn in Kelowna and not have to come to Vancouver, and so on and so forth. But all of these programs are not being just brought forward as interesting opportunities, but rather in a very close association of people like Larry.
Oh, sure. Yeah, so maybe I'll answer two questions that are somewhat related. So the first question is, how long do you think it will take to effect real change? So based on what we do know, I would say 6 months. So at 6 months, we actually do see significant changes in performance. So if we assess you today and 6 months later and you were doing the exercises, we would see quite a noticeable change, and I would assume you would too. But more importantly, with neuroimaging, we would actually we have seen changes. So both in brain structure, so the amount of brain in certain areas, as well as how your brain functions. But that's not to say shorter bouts are not helpful. So we also do know from other studies that a single session of either strength training or aerobic training can induce positive benefits that can persist for 24 hours. And if we actually collected blood from you, we can actually also see increases in growth factors. So I alluded to these factors in my presentation, but they really are critical in how exercise brings about really positive effects on your brain. So when you do exercise, your body produces more of these growth factors. They circulate, they cross the blood-brain barrier, goes into your brain, and induce very central effects. The other question was about strength training or weightlifting specifically, and that was, could you get benefit if you used your own body weight? So first of all, when talk about weightlifting, please don't think of it as, you have to go to the gym and lift the really super heavy weights and have to growl while you do it. We really are talking about, body weight and maybe a little bit more on top of that. Strength training has multitude of benefits aside from cognition and brain health. As you age, you tend to lose muscle mass, and that can affect your actual independence. If you can't get out of a chair or climb stairs, that's problematic for independent living. So strength training alone really is, I think, a part of healthy aging, but in terms of the type of training we have tested in our own lab, it is squats, lunges with light loading on top of that. We do use some machine-based protocols as well, but they're, again, not where you're, really loading and potentially harming yourself.
Thank you. I mean, as a family physician, I try not to use the word exercise because it conjures up sweating and smelly gear and hockey and stuff. And so I tend to use the word activity a lot when I'm chatting with patients. And I find the biggest bang for buck, and it's supported by evidence, is going from completely sedentary, doing less than 1,000 steps a day, to doing 1,500 to 2,000 steps a day. I wear a pedometer because I'm obsessive compulsive. And I like to compete with my sister, who's in Los Angeles, and my niece, who's down in Portland. And we compete on a weekly basis. I'm losing at the moment. But just doing a bit more can make an incredible difference. So whether it's actually doing some strengthening or whether it's doing some walking, and doing more will have an amazing effect on you. What I think is also fascinating is how it actually changes that. And that's the science part of me, being interested in how can activity actually prevent cancer, prevent diabetes, prevent just about every organ getting worse, as well as actually make some organs better. Yes?
I have a question for you, Martin. I'm going to pass this one on to you. Our questioner says, I heard that we should move from health care to sick care. What stops us from doing it now? I think doctors are not trained in health care.
Okay, so the quick answer is we are trained, but we don't always find time. So we train because I'm head of a department, obviously I'm deeply involved in the undergraduate and the postgraduate training doctors to be family physicians programs. We do emphasize the patient-centered approach and addressing health, what I was just talking about, healthy eating, activity, but dealing with the other stresses as well, poverty, environmental exposure, smoking, alcohol. We actually are trained to deal with that. We actually we test our students on how they can recognize someone who actually is being exposed to unusual social stresses and perhaps going off the beaten track and not looking after themselves. So we do address that. The trouble is that when we get into the emergency room and there are 14 people waiting for you, you go into a mode of dealing with the sick. I need to find out what's wrong and treat it. And it's really difficult to find time when you're looking after 1,500 patients in family practice to sit down with someone and go through what is troubling them, why they're anxious. Of course, if you do that, people feel a lot better. Absolutely. And our students know it, our residents know it, our doctors know it. And it's just finding time in the system, which is under a lot of pressure to do that. But, yeah, no, we strive to do that. And there was a wonderful article by one family physician in the BMJ. She came home and her son said, so did you save any lives today? And he had been watching House or something like that. And she would knock back and then said, yes, I did. And I talked to 3 people about stopping smoking. I talked to 1 person about alcohol reduction. I suggested activity to another 2 people. Yes, I save lives. So we try, and we need to be reminded to do more.
Well, and I guess I would just put the question back and put the question to the audience and say, if Martin talks to you about activity, do you go home and execute to actually change your behavior? If we sequence your genome and we find a modifiable risk factor, do you do something about that? That it's a team approach here.
It is. I mean, There's one thing I'm also interested in, which is the protein expression. So, you know, I have my genes, but I express some and I suppress others through what I eat, drink, how I've been, you know, what I've been through in my life physically and emotionally. That can be measured. And soon we'll be measuring protein expression, and then I'll go and say, oh my goodness, my brain failure has started to increase. The proteins that are more likely to be associated with brain failure are there. I need to actually go out and get more active, compete with my sister, and then see it actually drop. See those proteins actually start to reduce. That could be a massive trigger to more of us being active. If we can see, you know, waiting for your blood pressure to drop, or waiting for you not to have a heart attack. Well, you can't see that, but we can actually measure the proteins that are the cause of these diseases and say these are being altered. That could be cool. Ada, you have your finger up, and then Peter.
I was just going to say back to the question. I think it does make a difference when you have a personalized test or personalized information that says you actually are at risk, let's say, for a heart disease or something else. You can know intellectually that you have a parent who perhaps had problems. You know you should, but it's very different when you actually receive results that say you have a gene that predisposes you to a much higher risk of whatever it is, heart disease or anything. And so I do think it will make a difference to go forward.
Peter. Lightweight microphones. You see how they're doing their strengthening exercises already?
Yeah, this is actually it hasn't been brought up in one of the questions, but it's been sort of alluded to. And it has to do with, and this is something I'm going to. There's one of the points that Martin made to me a couple of years ago that had considerable impact, and that I think should be brought home in general. And it's the question of the number needed to treat. You know, we're all aware that we should. If you have high blood pressure, then you're often put on a particular blood pressure lowering drug, or if you have high cholesterol, then you go on a statin. But what you don't really, and that reduces your risk of a stroke or a heart attack by often a considerable amount. But all those drugs have side effects. But the number needed to treat is, to me, rather an interesting indicator here. I don't know if you want to follow up on that, Martin.
Oh, thank you. See how we work so well together. He lays it out and then drops the technical part on me. So number needed to treat for hypertension, mild to moderate with no other risk factors. I'd need to treat with an effective blood pressure lowering therapy about 120 people for 5 years to prevent one of them having a stroke. Now, wouldn't it be great if I actually had a genetic marker or a protein marker that helped me identify those who were going to get the bang for buck? Or weren't going to get the benefit from that drug? That would be even better. So I would not even put them on it and say, actually, you need to get active because the drugs aren't going to help you. I think that's extreme. There will be drugs that will help. But number needed to treat for putting a healthy 45-year-old man on a statin who's only got raised cholesterol. We're talking about treating 100 of them for 5 years to prevent one person having a cardiac event. You know, these are huge numbers, and maybe 10% of them will get calf pain or other muscle pain, and some of it quite bad. Again, wouldn't it be nice if I could actually do a test that would predict those who are going to get that pain and avoid giving it to them? It'll be harder for me to have a test that will say you're definitely going to benefit. But maybe I could measure the proteins, and after a month or two of treatment, I'd say your proteins aren't changing at all with the statins. I don't think we should continue this approach. Healthy eating for you, Mediterranean diet. Thank you. But numbers needed to treat demonstrate to us that 90% of drugs only work in 30 to 35% of people, broadly. Not the cancer, but everything lumped together, which is quite shocking. And now some really work well. Painkillers tend to reduce pain. But even then, strangely enough, the number needed to treat is 2 for Tylenol 3. So I've got to give 2 patients Tylenol 3 for one of them to have a 60% reduction in pain. So that's numbers needed to treat. Thank you for that. So actually for all of you, 5 minutes to go. Are there many international collaboration efforts? I thought that was an interesting question to sort of come towards the end. And you can talk about whatever you want anyway. But let's start from that end, coming this way. International collaboration efforts in your particular area.
I would have to say yes, given that I think we're right on the front edge of the curve here, and hopefully you've been given an idea that I think the storm is coming, and again, I can talk at it from a cost perspective, that there are more things than we can invest in than we have money to actually pay for what we can do, unless they're all fabulous and they're cost-saving, but that's not what we've seen. We may see that, but it's no different in Canada than it is in the UK, than it is in the Australia, and I have colleagues in multiple countries who are working on the same questions, because this really does look like it's the next paradigm, and the question is, trying to answer that question. What should we pay for as opposed to what can we pay for?
Yeah, so the answer is absolutely. There's a lot of collaboration generally in the field of cognitive aging. So we have, at UBC alone, we have ties with universities not just in the States, but also in Europe and Australia. And again, all in the vein of trying to figure out how can we all age well, both physically and cognitively. A piece maybe I should, if I could add, is that so there's been a lot of talk about the genomics as well as biomarkers and disease. The one piece that hasn't really brought forth is sort of the brain and how we map the brain. So similar to the extensive effort that went into mapping the genetic or genes, there's now extensive efforts in mapping the brain. So I had one question saying, how do you explain Stephen Hawking? Well, he's a variant. He's a visionary, but he's a variant. If you imagine, he somehow his genome has, or if the parallel of genome, but for the brain, allows him to do what he does. And we don't understand the brain at all at that level. So right now there's efforts, especially in the States, where there's a human connectome project, where basically they're scanning 1,200 young adults at this point in time with really advanced neuroimaging techniques. And what we're trying to understand collectively is how all these different wirings and connection contribute to the behavior and also neurodegenerative disease. So for those individuals who are great at math, just born with that innate ability, what is it about their brain that allows them to do that? And then similarly, for those who are predisposed to, certain diseases, is there some manifestation, again, within the connectome that we can map? And the future is, that, at least in neuroscience, is to connect the connectome with the genetic or genomic data and really allow sort of, I think, further advances in how we understand the connection between the genes and the brain.
Thank you. Briefly, Ada?
I've already mentioned that there is work going on. There's really an explosion of initiatives to capture data from human genome from populations. So I mentioned the President Obama, the 1 million genome volunteers, the UK 100,000, and so forth. But what's also going on, and Genome BC is very involved in this, and that is looking at the implications on society of all this genomic research and the discoveries. I have a couple of questions here about privacy, for example, and the sharing of data for research purposes, but what else how else might data be used? So that's a huge issue. We don't have time to talk about it tonight, but it's something that's being discussed widely, and people are very cognizant globally of what are some of the implications and how will we deal with the societal impacts to make sure people's information is protected, but also that we learn from all the data that's available, as well as what about insurance, et cetera, and all the other aspects around that.
Thank you. Bruce?
Just simply, yes, absolutely, everything is international, even with countries like Ontario. And so we work with all parties because this is both an amazing opportunity but also an amazing challenge. So you need the best people. There's always somebody somewhere who knows. You need the best programs to interact, and you need the best property or tools, technology, and be able to harness those in an economical way. So all of this is completely open international. and there isn't anybody that's involved in any of this work that would not be international. I do want to ask a question that could have come from my wife, but came from the audience. The person says, "I'm going deaf." Parenthesis, industrial exposure. Is it too late for rehabilitation? And that's what I thought this was from my wife initially. That she asks all the time why I don't listen. But so the answer is that even though there are many underpinnings, environmental, behavioral, and genetic, for various forms of deafness, I would say that there's astonishing improvement in hearing aid technology, in fidelity and size and cost, and there's a whole range of opportunities through the folks in the hearing world, otolaryngology and audiology. So the answer is never too late if you're just going deaf. So I would seek help.
No, it's appropriate we end with Peter, who's the father of the PMI, the Personalized Medicine Initiative, and has brought us all together and nurtured this baby. And Peter.
Okay, thanks very much, Martin. Just with regard to the collaboration aspect, I think as everybody along the panel here has indicated, this is front and center and central to everything that we do. I mean, obviously, we're a relatively small population in the context of the world, and so the extent to which we interact with and get best practices from the rest of the world, and then apply them to our community, so that we can then get to perhaps a better and we can compete more effectively with the outside world. Is a large part of what we do. I mean, this is a top of mind for sure. I was in England last week on a mission where we were doing exactly that. As Ida mentioned, there's 100,000 genomes sequence effort going on in England. How are they dealing with both the mechanics of that, the ethics, gathering the data, storing the samples? And also, interestingly, and it's something we haven't discussed tonight, how are they capitalizing on that information? How are they feeding the new industries that are arising from this? This level of understanding of ourselves is clearly going to lead to one of the biggest, if not the biggest, industry of our time. And so I think the opportunities are right across the board. And that we have to be really aware of all the efforts going on worldwide to make sure it happens here, and we don't get with that. We surf the tsunami of change, and we don't get drowned by it.
So we are very grateful for your questions. I think they've added enormously to the debate. They've helped us understand where your anxieties are. Which helps us then in either producing materials or going forward or doing our research or taking it into clinical practice. I want to thank all the panelists because they've been fantastic at responding so quickly to these questions and engaging with you. And thank you for coming.